As members of Carleton University, we're all here to help society in some way. Some of us apply our passions to study and improving the environment, technology society, and health. I'm here to apply my passions to improving patient care and quality of life. Specifically, I develop novel diagnostics and therapeutics to people with rare bleeding disorders. In this way, I'm helping to design the future of medicine. Bleeding occurs in three big steps. First, patients are injured, and then they bleed, and then they clot over 15 minutes, after which they can go get a pizza. Not everybody can do this effectively, though. In fact, 1 in 4 deaths worldwide are due to some sort of clotting or bleeding disorder, and bleeding disorders in general affect 1 in 10,000 people globally. It is these people I am trying to help. How can we help? The Goswami lab at Carleton University can produce small molecules that can act as diagnostics or therapeutics. Specifically, these molecules can attach to proteins that cause disease. By attaching to these types of proteins, our small molecules can both detect and deactivate the protein of interest. Using our artificial intelligence, we designed 23 small molecules to our protein of interest, a platelet protein that, when mutated, causes von Willebrand disease, which has no effective therapeutics nor diagnostics. Just for everybody to know, the target protein, my mutated platelet protein, will always appear as green in this presentation. I'll now show you 3 results that highlight the potency of one of these small molecules. First is this result in which, if we look at the pictures, on the top picture, there's what I call a synthetic clot. On the bottom picture with yes beside it, this synthetic clot is dispersed. The clot only becomes dispersed when I add one of these small molecules. And in fact, the quantification is just to the side with the red box outlining the highlight result, in that with no small molecule, the clotting measurement is around 6.5. With our small molecule, the clotting goes down to around 1. This is important because the disease I'm studying, actually clots are bigger and form easier. So we're looking to prevent that in this disease. And this molecule we developed can quite potently prevent clot formation. Next, in a different experiment, I incubated our green platelet protein that is mutated with its partner protein. And both of these, when these 2 proteins interact, they form these big clots. I then for 15 minutes, I incubated these 2 proteins, and then I separated that incubation into 2 equal tubes. To one, I added saline, to the other, I added my small molecule. The idea is to see what our small molecule does. What we found is that this magenta small molecule strips off our green target protein from its blue partner protein. And here's the actual result here. You can see that our small molecule reduces this protein interaction between green and blue by nearly 100%. It is quite potent, quite remarkably potent at that. I then, as a third highlight result, these are not all the results, but as the third highlight, I then demonstrated that our magenta molecule will specifically interact with our mutated platelet protein. And in doing so, it will, upon the addition of a detection reagent, wells in a 9/6 plate will glow yellow. However, if the molecule does not interact with the protein, as seen here, no yellow color will be seen. And this is, again, exactly what we see. If you look at the picture, the yellow glow is our target protein, the mutated platelet protein, and then our partner protein is on the right with no interaction. And again, this is quite a potent interaction. So this molecule has an extremely high affinity at 6.6 picomolar. The lower the number, the better. For comparison's sake, this COVID-19 human receptor interaction is 100 picomolar. So our molecule is extremely effective and has extremely high affinity to our platelet protein. Currently, we're testing the therapeutic candidate on patient platelets through our collaborators in Italy, New York, and Kingston. Pending these results, we're going to go into the mouse model in 2025, and we're going to further develop our diagnostic molecules with our industry collaborators throughout 2025. So in all, We are showcasing that our technology and these results to the world. In doing so, we're showing Carleton's capital advantage. Thank you very much.